Hi everyone, thank you so much for inviting me to present my work, Beta Spore Hygromorphs as a Novel Smart Biomaterial at this ACADIA World Workshop. I'm Emily Birch and I'm a PhD student as part of the Hub for Biotechnology in the Built Environment, which is a joint initiative between Newcastle and Northumbria University. So, a little bit of my background first. I studied my architecture part 1 at Newcastle University, and I was actually introduced to the smart material I'll be sharing with you today during my dissertation project. This project led to me receiving a research scholarship from the university and subsequently my PhD studentship. I'm now in my second year of my PhD at Newcastle University and part of the HBBE. HBBE has 4 main themes. I'm part of the Living Construction theme, which is a fantastically diverse multidisciplinary team of like-minded researchers, from microbiologists to architects and textile specialists, which actively promotes cross-field collaborations. My focus is in developing the potential of active biomaterials for application to the architectural field. This aims to push current static buildings towards a more organic, responsive and greener future by developing a new type of active biomaterial which can sense and respond to environmental changes with the end goal of a living architectural future. The PhD programme here at Newcastle University is typically 3 to 4 years, and we are actively encouraged to attend around 70 hours of CPD events throughout each year to ensure we're well-rounded researchers. These can be anything from presenting at conferences to ascending subject area symposium series to mindfulness and research skills workshops. Background and scope. So the drive behind this research is imperative. I'm sure a lot of you will have seen Sir David Attenborough's latest film and aware how critical it is that we make changes to our energy consumption and how we view finite resources. We really need to rethink how we are powering and maintaining our built environment, as currently buildings and the construction sector are responsible to 40% of the global energy emissions. There have been numerous attempts to lower the built environment's carbon footprint, but to date none have been inclusively successful. Space cooling seems to be the most difficult to lower consumption-wise due to the complexities of controlling relative humidity and condensation during the cooling process. So, I began to research current humidity control mechanisms, using what I found to produce a schematic, characterising them into active, hybrid, and passive humidity control systems. This highlighted that currently there are no commercially available passive but also effective humidity control mechanisms. So I turned to the natural world for inspiration. There is a naturally occurring class of material which is capable of responding directly to changes in relative humidity passively, called hygromorphs. Hygromorphic materials are those which are moisture sensitive, meaning they shrink and swell in response to water. This can be complete submersion or relative humidity depending on the type of hygromorph. These hygromorphic materials provide an active response to their environment whilst remaining passive in their energy consumption, something that we've not been able to achieve in the synthetic world yet. They are capable of utilising the tiny energies released during evaporation to fuel their shape change, rendering them passive on the architectural scale. There are many examples of hygromorphic materials within the natural world that you'll be familiar with, such as the pine cone in the video or the bacterial spores pictured at the bottom. These spores will become the foundation of our smart material development. So, having discovered a material capable of passage change in response to relative humidity, I began developing my research aim. It was really important that this aim was capable of changing perspectives of relative humidity and challenging the negative connotations that humidity within the built environment holds. The goal here is to not only provide a solution, but to engage users and develop their understanding of their direct impact on their internal environments. So, my aim is to engineer a creature capable of symbiosis and translation between humans and architecture by synthesizing a novel smart biomaterial from bacterial spore hygromorphs, utilizing understanding of passive nastic movement mechanisms and development of computer-aided fabrication techniques. This was then split up into 3 key objectives. The first is to investigate the microbiology of bacterial spores and understand their behaviour on a cellular level. The second is then to adopt a biomimetic approach using form and function understanding from plant biomechanics of passive nastic movement mechanisms. The third is to develop a novel fabrication technique capable of incorporation of a living active element into a living creature capable of complex interactions and shape change. While each of these objectives will inform on a specific level or hierarchy, for example, Objective 1 will be focusing on the cell or tissue level, Objective 2 will focus on the organ or organ system level, I will be referring right back to the cellular level on each objective to gain a better overall understanding of this creature as it evolves into its 4D physical realisation. So, this brings me to my methodologies. As each objective is set within its own discipline, I have had to adopt multiple methodologies to be appropriate at each stage. For Objective 1, I have followed a very classical scientific approach, utilising laboratory work to both develop and understand the B. subtilis spores at the micro level. To achieve this, I have self-studied certain areas of microbiology, so I had the relevant skills to produce a B. subtilis bacterial culture, monitor and regulate the growth of that culture, and then instigate the sporulation process at the correct point. This has involved learning key laboratory skills, including complex staining methods, as shown in the bottom of your screen. This staining process is vital to monitoring the sporulation process, as it stains the live bacteria red and the spores in a lovely green-blue colour, so you can see the transition of the culture as it undergoes the change from bacteria to spore. Having now validated the methodologies and protocols involved in producing a usable spore culture, work on enhancing the natural hygromorphic response of the spores will begin by altering the spore culture conditions ever so slightly to provide an optimum condition for the peptidoglycan production. For Objective 2, it was essential to gain understanding of the specific tissue architecture behind these movement mechanisms. Through literature studies, I was able to focus on 3 species of plant capable of reversible nastic movement, which is stimulated by a singular cellular expansion. This is directly translatable to the bacterial spores motion, so provide a potential biomimetic solution to amplifying the hygromorphic response. In-depth histological investigations of these plants has helped identify the specific structures within the plant tissue makeup which either aid motion, protect structures during motion, or amplify the shape change or speed of change. For example, the Venus flytrap utilises a bistable structure to allow it to snap from the open to closed position incredibly quickly. Isolating and understanding such structures provides a potential solution for upscaling and programmability of this micro-technology if synthetic fabrication techniques are capable of replicating them. So, this brings me to Objective 3. For this phase of research, a bottom-up material tinkering mindset has been adopted, asking the material what form it wants to take instead of beginning with a specific output in mind. Using the knowledge of the hygromorphic material from Objective 1 and incorporating the specific architectures understood from Objective 2, Objective 3 aims to combine all of this knowledge together into one creature, using additive manufacturing process to realise it in the physical world. From our knowledge gained at Objective 1, we know that the movement achieved by the hygromorphic material can be programmed directionally using the elasticity of the substrate material as a restraint. This relationship between elasticity of material and force output of the spores provides a baseline for such design experiments and tinkering. This process begins with investigations into flexible filaments. We have begun to push the limits of the material's capabilities, investigating print patterns and infill's effect on elasticity, the limits of material thickness and form, and the lack or use of supports. By understanding the limits of our printable material, we will then be able to feed that back into the design process to provide further parameters to aid our discovery of this new creature. Computer-aided design has the potential to evolve this creature beyond simple biomimicry and our own imaginations. Initial plant-inspired structures such as 3D printed mimosa leaf have been designed in Rhino using direct anatomical and histological studies of the plant. This just gives a superficial form, lacking in the specific internal tissue architectures which create motion. By using techniques such as 3D scanning, these biomechanical architectures can be further understood and visualized. While generative design software is mostly used with rigid product design and engineering elements, our hope is to combine the force output knowledge from the spores in Objective 1 with these visible structures within a generative design to provide a unique anatomy and biomechanics of our evolving hybrid creature.
So, results-wise, we have so far been able to achieve a pure bacterial spore culture, harvest and application of those spores so a substrate material for testing. These tests have shown that an increase in spore concentration leads to an increase in both deflection angle and force output. These responses were observed to be rapid, with a maximum shape change being seen within 3 minutes. They were also reversible and repeatable over the 20 cycles tested. This was then shown that actuators with the highest spore concentration were capable of lifting paper masses, as shown in the video. The mass lifted here is actually 1.5 times the actuator's own mass, showing a huge potential for force output. All of these observations show that the initial material prototypes are predictable, showing the potential for programmability. They can be calibrated as such to show a specific deflection at a known humidity. So, the aspirational outcomes for this research is to provide a material palette to demonstrate the capabilities of this novel bacterial spore-based smart material. With this, I hope to provide the technical and design foundations to inspire further development of this material towards the architectural application. Utilising this material palette to form a spectacle creature will showcase a wide variety of complex motions and interactions between user and environment that this material is capable of, whilst also providing an interactive and visual way of raising awareness and challenging preconceptions about our impact on the internal environments, hopefully contributing towards a more considerate and sustainable vision for our architectural future.
Thank you so much for taking the time to listen to my presentation today. I eagerly await your comments and questions. Thank you very much.